So, hello everyone. In today's session, we'll be studying about the bacterial transformation or DNA transformation. Okay, so here particularly we got one question that is related to DNA transformation in a bacterial cell. So this question I have taken from GATE 2017 question paper from microbiology section, and this is question number 17. To answer question, it is a numerical question. So it has been asked that 1 ml sample of a bacterial culture was serially diluted to 10 to the power 5 times and 46 colonies were obtained. After plating this diluted sample on an agar medium, the number of cells present per ml in the undiluted original sample were. Okay, so here it has been talked about the transformation protocol. So transformations are nothing but inserting the DNA fragment, or in most of the cases a plasmid, into the bacterial cell. So for that we have a generalized transformation protocol for bacterial transformation. So there what we do, we take a certain amount of DNA, mix it with the competent bacterial cells. So here competent bacterial cells mean that the cells which have the ability to easily take up the DNA. So some cells are naturally competent, but some we have to make them competent. So the most general method used to make the cells competent is the calcium chloride dependent method, where this divalent cation calcium chloride, it attaches to the around the cell wall of the bacterial membrane and allow the easy intake of the negatively charged DNA molecule. So that is what happens in a bacterial transformation. Then we give a short term period heat shock at 42 degree. That shock is to take up the DNA quickly by the bacteria, and then give it a cold shock. Allow the bacterial cells to recover by providing them some growth medium, and then we plate them on a agar plate which contains the certain antibiotic selection. So suppose my plasmid, I have in the plasmid I have a gene that is ampicillin resistance gene. So this, due to the presence of this gene, if this bacteria, or if this of the bacteria which has taken up this plasmid, if the on the plate you give antibiotic ampicillin, then also because of the presence of this gene, the bacteria is able to grow on that plate. So antibiotics are basically to kill the bacteria, but in our case what is happening, as we are introducing this foreign plasmid inside a bacterial cell, so bacteria become resistant to the antibiotic, and all the other bacteria which has not taken up this particular plasmid, they obviously don't have any resistance against that antibiotic, and that antibiotic gonna kill the bacteria which has not taken up the DNA, or those we can call them as a transformant. So non-transformant will not be grown and killed by the antibiotic action. However, the transformant cells will be growing on the antibiotic plates. So this is taken by the bacteria. Okay, and then when you plate it, you will get colonies. So these are the colonies or transformants which has taken up the DNA. Okay, so these colonies are also called as CFU. We can refer them as colony forming units. Okay, so in the question, it has been said that the bacterial culture was serially diluted. 1 ml of the bacterial culture was serially diluted 10 to the power 5 times, and 46 colonies were obtained, and after this they have plated it on a agar medium, and they are asking the number of cells present in the undiluted sample. Okay, So, talking about transformation, let's also talk about the transformation efficiency. So it refers to the ability of the cell to take up the DNA. So once you um, get the transformants, what you can also do is to do the calculation of the efficiency of the DNA. So accordingly, next time onwards, you can add the appropriate amount of the DNA, and then you should know that what will be the expected outcome, how many colonies you will get, and something like that. Those calculation becomes easier. So transformation efficiency is nothing but the efficiency of transformant, and it is measured as the number of transformants per microgram of DNA added to the cell. So this is the formula for transformation efficiency. So it is CFU uh, per concentration of DNA plated. Okay. So CFU we can count like on a plate how many colonies I am getting. Okay, so if I am getting 100 colonies, so 100 will be my CFU, because bacteria we know we cannot see them on eyes. So whatever these colonies we are seeing, that like one bacteria is dividing so much and making a colony which is now visible to our eyes. So that is a colony forming unit. Okay, a colony, one single bacteria makes the entire colony, so that is one colony forming unit or one transformant. Okay, now this is the concentration of DNA plated. Okay, so you take certain amount to transform uh, or you add certain amount to transform the bacterial cells, but you need to know how much you are actually plating on the plate. Not in most of the cases you are not going to plate the entire thing. So to know the concentration of DNA plated, the calculation is amount of the DNA transformed, like how much you have taken initially, how much you have plated, and the volume at the time of recovery. As I have mentioned that we give some medium for the recovery of the cells, the final volume at the time of recovery. Then our calculation becomes, so this number of transformants per microgram or transformation efficiency will look something like this. So number of transformants, which is CFU, number of colonies upon microgram of DNA into volume plated, and this final volume at the time of recovery. So this is the calculation. Okay. So with the example, if I'll say you, so for example, you got 350 colonies on a plate, on an antibiotic plate after the transformation. How much you have plated? You have plated only 100 microliter. And how much the total suspension? It was 1 ml. Okay. So 1 ml was the total suspension volume. That means at the time of recovery, you have added 1 ml media. So that is the total 1 ml amount. Okay, and from there you have plated 100 microliter. And how much you have added the DNA? So that is 200 nanograms. So now you need to know how much you have actually plated. Okay, so yeah, so that also you need to convert into certain volumes. Okay, so all the volume should be in ml, DNA should be in microgram, because we are talking number of transformants per microgram, and this is will be as it is CFU. Okay, so the plated volume is 100 microliter. That means 100 microliter means okay, this is a wrong calculation. Yeah, it will be 0.1 ml. Okay, there also it will be 0.1 ml. So the total suspension is 1 ml, and the amount of plasmid is 0.2 microgram. Okay, so the transformation efficiency will calculate CFU into final volume upon amount of DNA added, amount of DNA plated. So after the calculation, it comes like this. Okay, so this will be your transformation efficiency after solving this. So this is how you can calculate the efficiency and everything. So next, if I'll see in the question, so in the question what they have did, they have diluted. So this 1 ml, which I am just saying that the recovery volume, so this also they have diluted 10 to the power 5 times. Okay, so this is 10 to the power, so this is 10 to the power 0 dilution, this is -1, or you can say, and this is -2, -3, -4, -5. So this is 10 to the power 5 times diluted. Okay, so here so if it looks something like this, so if you'll play it from here, you'll get something like this, a whole lawn. If you play it from here, you'll get something like this, a less dense, because the number of the bacterial cells in the suspension are becoming less and less. Then third dilution, you'll get even less colonies, and then fourth you'll get lesser, even lesser, and then very less. Okay, so you got only 2 colonies. Then you can cross back, like calculate it back, and then get to know how many colonies are present in the original sample. Okay, so this is also a way to do the calculation. So if you are getting 2 colonies here, that means originally you have 2 into 10 to the power 5. Okay, so if you are getting, and if you go just 1 dilution back, that means you have 2 here. This is 10 times lesser diluted, so here you will get 20, here you will get 200, here you will get 2000, here you will get 20,000, and here you will get 2 lakhs. Okay, so like this. So similarly, if you are getting 46 colonies, so you will be getting 46 into 10 to the power 5. This will be the answer. So these many colonies will be present in the original sample. Okay, so this is easy, very easy question, very direct. Only thing you should know the basics and the background and how the technique is performed, so that you will be easily understand what has been asked in the question. Okay, so thank you. This is my NPTEL reference. You can refer to this to see carefully how the transformation actually works, what you expect, how you perform, and everything. Thank you.